All right, welcome to the review for chapter 4. Let's run through it. So chapter 4, we talked about bacteria and archaea and kind of the differences in their structures, mostly about bacteria, of course, and that's what this course is mostly going to be talking about is bacteria, so that's okay. Bacteria and archaea, they do not have any membrane-bound internal structures. They are eukaryotes. We are eukaryotes, true karyotes. That just means we have true membrane-bound nucleus. We have true membrane-bound organelles that perform functions within our cell. They don't. Everything's just kind of going on in their cytoplasm, which is the goo inside of them, and along their inner cytoplasmic membrane. So cytoplasm being the cytoplasm side of the cell. So that's what contains everything is the cytoplasmic membrane. All right, there's different shapes of cells that we had talked about. The shapes, the biggest two basic ones were cocci, which are the round ones, and bacilli, which are the rods. Those are the two big ones I really want you to be familiar with. And then we have the curved ones, which could be like spiral shaped and comma shaped and stuff like that. But definitely be aware of the cocci, the round ones, and the bacilli, the rods. And then the different arrangements of the cells. That's just based on how the cells are associated with one another when you look at them in the microscope and when they're growing. So staphylo is going to be referring to grape-like or irregular clusters. Strepto is going to be strips or chains, and diplo might be pairs. There's other arrangements as well, like tetrad, which is going to be just 4, or sarcina, which is like a cube arrangement. For the rods, we have the palisade, which is like a fence. So they're just attached at certain ends whenever they were dividing. And that's what causes that to happen. So that's the arrangements that you can see with regards to your bacteria. How do bacteria move around? They move via flagella. So it can be flagella on the outside, or it can be essentially flagella on the inside, which is the axial filament, which is kind of spiraled around inside the cell and can twist and allow the cell to move around that way. That's the way that bacteria move. That's the only way that you guys need to worry about, okay? If we're talking about fimbriae, if we're talking about pili, these are for attachment. Nanotubes or nanowires, those are for moving like molecules like nutrients and other like electrons and stuff in between cells, so super tiny things. Whereas the pilus is actually used not just for attachment, but also used for sharing like genetic information, like a plasmid across from one cell to the next, for example. So that's conjugation whenever that's happening with the pilus. The sex pilus does that. Okay, the flagella themselves, though, if we are a motile organism and we have flagella, they can be arranged around the cell at a polar configuration, or they could be peritrichous. The polar configurations are going to be amphitrichous, so that's at each end, monotrichous, one flagellum at one end, or lophotrichous. Lophotrichous has to do with a tuft at one end, so. The capsule layer, that's a protective layer, as well as a sticky layer on the outside. So it's part of one of the kinds of glycocalyx that which is a sugar-based coating on the outside of bacterial cells. Slime layers are loosely associated with the cells. Capsules are more tightly associated. These structures are going to allow them to stick to surfaces that can allow for bacteria to stick to the surface to one another and then grow upon one another, and they can even kind of talk to one another and let them know how the biofilm as a whole is doing, and that is called quorum sensing. The cell envelope is the entire outside enclosing layer. It's going to include the cytoplasmic membrane, the cell wall, and the outer membrane if you have one, right? So right, so the cell wall, so let's just talk about that real quick. That is made up of peptidoglycan, glycan. Every time you guys hear about that, I want you to be thinking of like sugar. So sugar-based. So some sugar-based structure, repeating subunits of sugar-like structure of NAM and NAG. So that's N-acetylglucosamine and N-acetylmuramic acid, repeating over and over again. And they are kind of, those chains of those are linked together using a peptide cross bridge. So short peptides of amino acids linked together. And that's going to create the peptidoglycan sort of scaffolding. It's very thick in the gram positive. Also, the gram positive has, so it's at the thick cell wall that is maintained by using the lipoteichoic and teichoic acid in the thick cell wall. Whenever it is dividing, it helps maintain that cell wall as we're making a new cell. So that's what that stuff is used for. It's only found in gram positives. Gram negatives, they have a thin wall, but they have an outer membrane. And in their outer membrane, very importantly, is a lipopolysaccharide. And like it sounds like, we've got sort of a lipid portion and then sugar portion. And then basically this structure is associated with endotoxin, which is something that triggers fever as well as shock response during infection with organisms that have lipopolysaccharide or endotoxin. And that's always only going to be gram negatives. You're right. Yes. Gram negatives. It's going to be gram negatives have endotoxin and it's dangerous. So then we had the acid fast bacteria. So they have mycolic acid, which is a waxy substance that provides protection and survival in harsh conditions. This is the Mycobacterium species. It also includes nocardia, but I don't really need you guys to know that. I really want you to be aware of the Mycobacterium species, like Mycobacterium tuberculosis or leprae, that are disease-causing organisms. I would be aware that this is an extremely important aspect of these organisms because that acid-fast stain is used all the time in hospitals to diagnose tuberculosis. So be aware of that. Know why it's done. Know what's going on chemically with that. And understand what that means to be acid fast, means to have mycolic acid, means to have that waxy coating, means to be able to be resistant to certain conditions that normally you could, get rid of an organism. Then we have the, right, we talked about, so mycoplasma organisms. So mycoplasma, they just don't have a cell wall at all. So they have a lot of different shapes of their cells. So they're very pleomorphic as well. So pleomorphic means they're not coccus, they're not bacillus. They have all these different shapes that you could see from that one species. Next, we're going to talk about the plasmids. Plasmids are small circular DNA. They're not essential for survival, but can code for virulence factors. A virulence factor is anything that allows that bacteria to gain foothold and grow and become a pathogen and cause an infection in the host. So anything. So it could be coding for a toxin. This is common, coding for a toxin or antibiotic resistance is also very common. And again, we can transfer plasmids, which are small little circles of DNA. They're small. In between the sex pilus, between bacteria to bacteria from the same species to same species. That's how that works. And so they can communicate and transfer antibiotic resistance that way. And it's a useful trait for them. We can hijack the system as well. We can take a plasmid and put our own gene of interest in there and put certain triggers associated with it that allow us to produce proteins of interest like insulin or something like that and cause bacteria to be able to produce, human proteins or whatever. It's a little more complicated than that, of course, but that's the idea. And that is how it's used in genetic engineering. So that's a useful tool, super useful as far as research science goes. Plasmids, again, I wanted to reiterate, they are in a circle. They are double-stranded DNA, regular DNA in a circle, tiny. The chromosome of bacteria is also in a circle, but it is much larger than the plasmid. Okay, so be clear on that one. All right, next we have the endospore. Endospores are formed by bacteria in harsh conditions, usually where they are lacking like nutrients, often the carbon or the nitrogen type nutrients. So they're lacking in the nutrients, or they may be getting dried out or something. So they produce endospores as a protective to protect their DNA, their genome, right, in harsh conditions. They can survive a lot of conditions like this. They can survive things like radiation and heat, freezing, and all sorts of stuff, pH changes. chemicals, all sorts of stuff that other bacteria might be exposed to and die, the endospores can survive through. So very hardy, very hard to get rid of. So that's the endospores. That's produced by the Bacillus and the Clostridium species. We talked through how endospores were made. The process of going from a vegetative cell, which is the typical metabolically active normal bacteria, going from that into endospore. That process is called sporulation. The cell that is undergoing making an endospore is called the sporangium. And then if we have made an endospore and we have favorable conditions and we want to come back out and be a vegetative cell, the process from going from endospore to vegetative cell is called germination. So those are the terms associated with that. Then we did talk about basically classifying organisms. We talked about Berge's manual a little bit, and identifying species based on a lot of different characteristics, whether it's phenotypic like biochemical tests or genotypic based on like gene sequencing. And then we have categorization here of organisms based on their cell envelope, essentially. So the gracillicutes are the gram negatives, the firmicutes are the gram positives. Remember that the thick cell wall, the thick cell wall made of peptidoglycan, that thick peptidoglycan layer is firm. So that's the firmicutes. And then we have the tenericutes, which have no cell wall. And then we have the Mendozicutes, which are archaea, archaic being old, my old boyfriend, Joey Mendoza. So then if I have those two knocked out, the firmicutes and the Mendozicutes, gram negatives start with G, that leaves me with gracillicutes, and then the last one is tenericutes. That's going to be your no cell wall. So that's how I remember that. If that works for you guys, I don't know if it does or doesn't, but that is the chapter 4 review. And I will see you guys for, I guess, the next one. I guess that's chapter 5. I'll see you later.